No, I don't have control. Okay. Who's actually the host? Of course, I'm not. Anyway, I'll go on. Okay, he spent 5 years at Penn. He moved to Harvard in 1999. He's primarily a physicist, but he's also a biologist and an engineer and an entrepreneur. He started at least a dozen companies throughout the world. They typically do things like droplet microfluidics to make things from perfumes or do sequencing. When you look him up in Wikipedia, it says he's best known for, and then there's a list of 16 different things. But the thing that he's most famous for is he never sleeps, and somehow he's figured out a way to do 120 hours of work each day. He's a member of the National Academy of Science and National Academy of Engineering, and despite having a group that numbers typically something like 100, he still found time to do things like develop and teach a course, Physics and Cooking, which for years was the most popular course in the whole university at Harvard. And what he's going to tell us about is something new today. It's dynamic speckle holography, and that may be the next thing that you're going to find in physics and biology labs around the world. Dave?
Great. Can you see my screen?
We can see. Yeah.
Yes.
Okay.
Great.
Let me just make sure. Hold on for a second. I just want to get my hand working. Okay, good. So I'm really, it's a pleasure to see you all. I really wish I could actually be there. Alexander and I sort of planned it out that maybe we would work, but it didn't quite. Sounds like it came pretty close, Alexander. It was a good try. I want to tell you something that I've never given this talk before, and I literally was putting slides together 2 minutes ago, or 7 minutes ago, because we started, but 2 minutes before. And so it's all new. But it seems I had to give this talk at NYU because Dave and Paul are there. And, you know, I remember, you guys remember when we did DWIS, how long it took to name it? I think it took us longer to figure out how to name it than how to do it. So we had the same problem here. I wanted to do something like that, and this is really dedicated to you guys. It's sort of the same thing. It's really the work, though, of Stefano, who was a student of Luca Cipolleti. Luca was a student of Marcio Giglio, and Marcio was the master of speckles. And Luca became a master of speckles, and so did Stefano. And this is really about using speckles. And it's called dynamic speckle holography. I want to show you. It's a technique that's it's got a lot of similarities to what we did with diffusing wave spectroscopy, but it's something different. It's all really Stefano's idea. And we've just had a lot of fun the last couple of years, first developing the technique and then learning about something that, again, is old things coming back new, and that's colloidal gels, and understanding a whole bunch of new things about them. And I want to tell you about that as well. The other person who was very helpful was C.G. Sun, one of the students in my group, and we have this long-standing collaboration with Li Zhishao from Chinese University of Petroleum in Beijing, and understanding porous media, which the colloidal gels mimic that. He's been very helpful for that. Okay, so here's the question. This is what we're really trying to do. Imagine you have this massive field of view and you want to see little tiny motion in that. So this is Stefano's little cartoon, and you want to see, what do you call them? These bugs, ladybugs, way far away. How do you do it? Well, you could look at it with a microscope, right? And you can image what's going on. You can image the bug and you can watch how it moves. And so the standard way of doing that is you image it and you watch it move. But as you get smaller and smaller, and you have to get smaller and smaller if you have this large field of view, you lose your resolution. So the question is, how can you measure something where you have the sensitivity of motion that's different than the resolution? Now, why is that? The reason is that if you think about it, whenever you're measuring motion, PIV, all kinds of anytime you measure motion, what you do is you look at some feature and you measure the change in the feature. So you always have to somehow resolve that feature and measure how it changes. And if you think about PIV, all of these things, it's all the same. So your sensitivity to the motion is related to your resolution. And you can't break that. It's inherent in the way you do the imaging. So the question is, can you somehow have sensitivity to motion that's much greater, but still see over a very large field of view? Can you break this coupling? And this is what I'm going to tell you about how you do this. So we're going to look for very small displacements, really, really tiny displacements over large fields of view at as high a rate as we can. And we're not going to do it. We're going to not try and resolve features to do it, but somehow do something else. And that's what dynamic speckle holography is. And this really is the invention of Stefano. And so what it is really simple in the end. It's taking advantage of interference. So interference is, we know this, everybody who's done dynamic light scattering knows it well, but for those of you who don't, imagine I have two particles and they're scattering light, and I don't try and image each particle, but I just look at the scattered light. Then when the particles move, the scattering changes. So you can see the scattering field from each of these particles is changing in time. And if I look at the total scattering, which is the coherent sum of these things, the intensity changes, the intensity fluctuates. But why does it fluctuate? It fluctuates because the particles are moving relative to one another. So the interference pattern between the two particles is changing the intensity. So I'm not resolving the particles. I'm just looking at the scattering of the particles. I'm looking at the changes of the intensity. And so if you see random fluctuations, randomly moving particles, it's because their intensity is changing over time. But we know how to take advantage. We know how to understand that. That's just looking at the relative motion, and we calculate correlation functions. And that's what diffusing wave spectroscopy is about. That's what dynamic light scattering is about. You're just calculating relative motion of the particles, how it changes the scattering, the intensity of the scattering. And we know how to do that analysis. So if we could somehow combine this with the large field of view, we could decouple the resolution of the objects that we're looking at and just look at the relative motion of them. And that's just looking at the interference patterns. So here's how we do it. This is a specific example. I'll tell you the example in a minute, but imagine this is our sample, and we do essentially a light scattering experiment. So we shine a laser here, and we look at the scattered light at some angle, but then we aperture it down and we image it, and we image the speckle. So we image the whole speckle pattern. So what this is there's a chamber in this way. We have fluid flowing in this direction, and it's just simple Poisson flow. We could measure the velocity. We could measure everything from doing that just by imaging the particles. And we could image the particles. We could image them and do PIV. And we can do all of that. But instead, what we do is we image the speckles. And I'm going to I think if I play this, I'm going to show you a movie. These are the speckle pattern. This is the speckle pattern here, and it's playing over and over again. And let me see, can I get this one? No, I can't. Somehow this should, if I look at this, it would also fluctuate as well. But I'm just looking at the speckle pattern, and now I have to analyze that speckle. And let me see if I can do that. Okay, so I apologize. Oops, sorry, I don't know what happened. I apologize for the way I copied this. It's not perfect, but it doesn't matter. It'll get me the things I want. What we do is, this is the same thing that I showed you. These are the speckle patterns. You can see this is the speckle pattern from the walls of the sample. This is the speckle pattern from the flowing particles. And we can analyze when we have the wave vector, the scattering vector, in the same direction as the motion. We see this set of correlation functions. All we do is we take speckles in some small region, some little region like that, and we calculate the local correlation function. So we don't, although this is a stationary system, so we can just do the normal light scattering that we do, we don't have to. If we look around at a few speckles and average them together, we can see the correlation. And we can see this. Notice also that you can see some little fluctuations here, right? In each case, there's some oscillations. And so how do we really understand that? Well, what we do is we remember that, let me see if I can go to the next figure. Yeah, here, where we remember that it's Poisson flow. So even though we're averaging, we're looking in this direction across, if we average over the whole volume, some of the particles are moving faster in the center, slower on the sides. So there's relative motion, but this is really highly correlated because they're moving really relative to one another. And so when we look at the actual motion, you can see that there's correlations. And we can actually calculate this. And let me go to that, back to that other slide here. Yeah. So we can calculate this, and we can scale all of the data together. Here, we're just scaling about by the velocity. So that's this decay. And when you do that, you see that even the oscillations scale together. And that's because we're moving at different speeds, but now we're taking slices at different regions, and they're moving at different relative amounts. So, you know, the particles in the middle are going so much faster than the particles on the edge. It's just it's a parabolic pattern, and this black line is just calculated what it is. We can actually do a lot more. You can put the scattering perpendicular, so the scattering vector is in this direction, and then what you do is you watch the speckles move through the beam, through the region that creates the speckle. It just moves coherently. And so that's a way of, when it's really moving faster, you can still do it. That's these data here. And in fact, even here, you can see there's a small decay. And when, here's the small decay. What's happening there, that's when we're looking at, even the velocity gets really, really, really slow, then we can start seeing Brownian motion. So we can see all the things that we would see with PIV and dynamic light scattering just by doing the same speckle imaging. There's nothing really profound about this. It's what you would expect. It's easy to understand. It's just now we're taking advantage of imaging. But what we've done, because we're imaging, is we've separated the motion of the displacement of the particles, the actual dynamics of the motion, the displacement, from the field of view. We no longer have to actually resolve things. We can just take advantage of the scattering. Now, we call it holography because we don't have to do it. We don't have to do just one direction. Here I've just put my scattering vector in one direction, but we could do in many directions. And let me show you some different systems where this really becomes essential. So now I want to change my discussion. I want to talk about a different problem, but a problem where this kind of behavior is just perfect. And so what it is, it's back to a simple system, and this is a colloidal gel. So we take colloidal particles, we aggregate them, and we form a gel. You can think of it like mud, like paint, some kind of fluid system of particles that is starting to form a solid, and then we dry it. We just allow air to come in and we dry it. We do it in a controlled way, and we watch how the cracks form. So the cracks that form, and the cracks are induced by air being injected in, or actually the water is evaporating and the air is displacing the water. It's really sort of an invasion effect. It's like invasion percolation in porous media, and cracks develop, something that's been widely studied. This is really not so different than the cracks in mud or in clay, or even the cracks in the window. It's the propagation of cracks through the system. And we can look at it when we do air, but I'll show you we can also induce the cracks by pushing a fluid through it, and that's where it becomes close to porous media, because we're looking really literally at hydrofracking. So how do we do this? So let's see, this is the same, yeah. So we also can do it by, instead of allowing evaporation, we can actually inject a fluid and we can develop cracks. We can inject several fluids from different regions, and we can watch the interaction between cracks. We can induce a crack in one place and then watch the interaction between the cracks as we induce it elsewhere. And we can do this with some control rate. We can measure we can instrument it. We can measure the pressures. At the same time, we can measure the mechanical properties of the gel, because we're just forming a gel. And so we can do rheological measurements on the gel as we form it. And the moduli are orders of hundreds of pascals to kilopascals, depending on the volume fraction of the sample. And we can vary the volume fraction, so we have a wide range of systems that we can study. This is a typical thing, the pressure goes up and then the crack nucleates, so we can follow this. I don't want to go into the details of that behavior. I'll come back to just using the numbers, because I need the numbers, but I want to understand something about the motion. So let me just show you the kind of motion that you see, and then I'll show you how we can use dynamic speckle holography, DSH, to actually study it. So here's just two cracks that are interacting, and let's just watch them. I'll play them. This is slowed down, the whole thing taking about an hour. Oops, sorry. I touched my pen, and let me just go back and start it again and watch them. They're approaching one another, and you see this one bends over, and then this other one bends in this direction. So this one goes this way, and this one goes this way. And so what's going on? Somehow this is going through a solid. Somehow the strain fields in the solid must be telling us why the cracks are moving the way they are. And so we, for example, just by looking at them, we can plot the direction and the acceleration, and the arrows show that. And you can see that they move in a different direction, and they speed up, and they know the effect of each other. They're coupled to one another. The only way they can be coupled is through the mechanics of the gel that surrounds them. So how can we understand that? Somehow what we have to do is measure the whole strain field of the gel. And that's where DSH is perfect. We have to measure very small motions over a large field of view, and the motions are all coupled together. So we want to see the correlations of the motions to understand the way that behaves. So let's see, what is this? This is the same thing. Sorry about this. Yeah, so how do we do this? So we do the same thing. We do dynamic speckle holography. This is playing a motion. This is, come on. Oh, this is starting the cracks. And now you can see the speckles. I hope I can see it perfectly on my Zoom. I don't know if you can see this when it goes over the internet, but if you can, you'll see that there's some motion of the speckles here, but not so much here. You can see clearly that the crack is moving just by analyzing the speckles. So we can do that, and we can measure the rate of decay. And here we just do a color plot, the speed. You can see the crack now very clearly. One thing I'll point out is that there seems to be a certain symmetry, but that symmetry is more defined by the scattering vector necessarily than by the the physics of the crack, and that's because we're just imaging in one direction. So what we really have to do is take a second laser and image in the X and the Y directions. And we do that. I'm not sure I have a movie here that, well, it shows how you do that. What we do actually is we have one detector, two lasers. I won't go through the details of the geometry, just the detectors are fairly expensive. So we have, we split the laser beam and then we chop it and we go from one to the other and we just take the combination. And now you can see here where I'm showing the displacements and the magnitudes, the speeds, and the symmetry now is really around the crack itself because we're getting both the X and the Y directions when we do this. So we have the two scattering and we can see both of them like this. Okay, so now if I actually make a plot of the motion of the crack, this is the motion of the crack, and you can see that we can measure the whole strain field all around the crack as it's occurring in time. We can watch it in time, we can watch the strain field, and now we can go back to those same images that I showed you before. And well, let's see, this is the same thing. This is just looking, again, doing the same type of cracks, but now watching the displacements as a function of time. These arrows are a function of time, and you can see the way as the cracks overlap, you get displacements in the middle. So let's look at this. We can go back here and now we can measure, we can use DSH to measure displacement fields as the two cracks are propagating, and you can start seeing how they're coupled and how they're coupled to one another. You can see the motion here, the motion here. We can go and deconstruct this. Here's just the divergence of this. And you can begin to see what's happening, why there's a coupling and why there's a motion. It's the motion here is because of this long range strain field. It's pulling the crack in this direction. And okay, we can go and analyze this. I'm happy to do this. We've done that sort of carefully. You can understand this. This is perhaps a good way that you see it, that the reason that that it goes perpendicular is because we have to pull the crack apart in this direction, and the crack here has loosened the solid here, so it can only pull in a direction that's the way it pulls is perpendicular to this growing crack, and that's why it's changed in this direction. And then because of the presence of this crack, this side is stronger than this side, and that's what pulls it off in that direction. You can start analyzing this and seeing this because you can now measure the whole strain field across the whole sample and understand it. Let's see, this is just looking at it in detail. I don't think I'm going to belabor the point. We looked at it carefully to understand the strain fields. I'm just going to skip through this and try and go a little bit farther. This is, this is looking, and we can see this. Now this is just doing the same thing. And we wanted to understand more. This is actually injecting a fluid. We wanted to understand more. How can we understand really what the nature of the fracture that we're inducing? So here's just a single crack. What can we understand about its nature? So we do the same correlation function, and this is what it looks like. You can see that when we use both directions, there's clearly something that goes perpendicular to the crack. That's what we expect. It's not changing its direction because there's only the single crack, and it's moving. And again, we just analyze the speckle, and we literally take a region like this little box, and we do the correlation function in each box. And from that, we can get a map of motions, a map of velocities, a map of displacements as a function of time. I've said this. Here's some data of a crack. This is like looking at one of the cracks. It's moving in this direction, and this is the displacement field, and you can see the direction, you can see the magnitude. And so now what we do is we just take a line across here and we plot what the displacement is as a function of distance. And this is what we get. The displacement as a function of distance from the crack after the crack has passed through, what we see is that there's an exponential decay, and the decay length is a few centimeters, but there's additional motion here. Let me see if I have something that explains this. Okay, so let me come back to this. The reason that we have this kind of motion is that, remember, what we do is we have a gel between two windows, two plates, and we're looking this way, and we're inducing a crack going into the board, and the crack actually looks like this. And so the motion, most of the motion is in the center. The gel is actually stuck to the windows. So most of the motion is in the center. We get this bulging out. And if you calculate the kind of behavior you expect to get, what the fall off in the displacement far away over here, you expect to see something that's exponential. You actually get the Poisson ratio from the decay length, and the Poisson ratio is very close to one. It's almost incompressible, but not completely incompressible. But notice that there really is something else going on here. And if we do this and we take the data here and we just calculate it, we get this green curve. We get a second exponential behavior. So there's something else that's happening right in here that is different. And notice that the arrows are not pointed away from the crack as they are away from here, but they're in different directions. There's some other kind of motion that's happening right in the middle of the crack. So how can we understand that? Right near the crack, how can we understand that? So to do that, we add the third dimension. So up till now, if we were doing it before, we had X and Y, but now we also add a Z component, and we do that by essentially measuring in the scattering, in the forward scattering, the back scattering, with a scattering vector that's very close to the X and Y plane. So the difference is just the small amount in the Z. And actually what you have to do is you have to make the two measurements and subtract to get rid of the X and Y component and just get the Z component. But we can do that, and we can measure the Z component as well. This is just showing how we get the Z component. It's not as sensitive, but it's good enough. And so now here, this is a full three-dimensional situation. This is this this interacting one, and you can see that there's the height measurement is given by this, and we can go back and we can look at these data, and this subtracted data, this blue data, is really from the Z direction. So this additional motion, why we're getting it is that we're getting additional motion, but we're also getting a large component of motion in the Z direction, the up-down direction. And how far does it extend? Well, the thickness of the sample is something of this order, so it extends roughly in the thickness of the sample. So what's that telling me? It's telling me that far away from the crack, it's moving only in this direction. It's being forced out, but near the crack, it's also moving in this direction, And you can see the analysis of the shape of the correlations when we look at the Z correlations. The analysis of the shape tells me that it's moving in some random way. And the reason we say it's moving in some random way is it's moving up and down. So really, what could be causing this additional motion in this direction? And so I'm going to speculate something, and then I'm going to go and look for that behavior. The thing about this sample is that a gel like this, we know, is actually under internal tension. And I'll show you evidence of that, why it's under internal tension. There's some rheological evidence, I'll come back to that, but it's pulling on itself. And if it's pulling on itself, it's going to pull to wherever it's stuck. So here, it's stuck to the gel deep into the sample. But here, it's stuck not only to the gel deep in the sample, but also to the walls. It's stuck to the walls. So here, if it's being pulled apart, then not only will it move in this direction, but it will move up and down because it will move to the nearest walls. So we'll see that effect. Here, it's moving to the walls, but there's just balance, and so it only can move in this direction. It doesn't move to the wall. It's not fracturing. But here, for something of order the size of a sample, you would expect to see some additional Z motion. Well, let's go and look more carefully at this. So the first thing we did was now look at the nature of the crack. And now I'm going to play something. I'm going to slow things down, but I'm going to look with very high resolution. I'm going to image it, not only do the speckle imaging, but do the actual imaging, because you see something quite interesting. When you play this, what you see is, notice that you see two effects. Let me wait till it stops and I'll draw on. You see two effects. You see this effect, and you see this effect here. This is air, so this is where it's drying. But you see a line here that moves with the crack. And if you look at it more carefully, what you see that this is the gel, this is air, and this is actually water. So you're drawing it and you're drawing water up, but the fracture, the tip of the fracture is always deep into the water. So it's a waterfront that's moving ahead of things. Let me see, what do I have here? Well, yeah, here, it's the same thing. And let me just see if I play this. Yeah, we can measure this front. This is water. This is air. This is an interface. We know the pressures, right? Because it's this way, that means the pressure here is less than, this is P water, is less than P air. We know that that's the case. There's an osmotic, there's a Laplace pressure that's due to this front. Let's look at this. Well, okay, we can calculate what the Laplace pressure is, of the order of a kilopascal, and that just happens to be of the order of the pressure of the stress that we know that we see that it breaks at. If we make some stress strain measurements, we find that this is some rheological measurements, just measuring stress and strain. We find that at 10% or 20% deformation, it's about of order kilopascals where it breaks, and that's where this is pulling apart, but it's breaking up here. So how do we understand that? Well, let me just play it again. This is a different thing, and here it's moving. You can see the water is always pushing ahead. And now I'm going to overlay some spheres, some circles, and you see that they're always the same value. Whenever this crosses, it's the same value. So the pressure here is the same. The pressure difference is the same. So how do we understand that? Well, we can look at the stresses. The yield stress is for this particular gel. Here we're measuring it. This is the yield stress. It's something of the order of 480 of order kilopascal. And it turns out the Laplace pressures, whether we're injecting a fluid, an oil, or air, are about the same. So what's happening? Well, let's look at the pressures. So I said that the pressure of the air is the higher pressure. The pressure of the water is lower. Here, far away, you must be, again, at atmospheric pressure. This is atmosphere. This is atmosphere. So this is drawing the water out. The pressure is drawing the water. It's a lower pressure. It's being drawn out. And somehow the gel is pulling itself apart. And the reason for that is it's under this internal tension. So we're pulling the water out, and that allows the gel to pull itself apart. So the gel is putting its own force on it here and here. And that's why the tip of the fracture leads where the water is. The gel pulls itself apart because of the internal tension. So let's see if we can understand what the physics of this is. So now we go back and we look at the behavior of gels. Okay, so this is a series of gels that Stefano made, and he measured the rheological properties as a function of volume fraction. The reason we do it with the system is that we can control the rheology, we control the mechanics, we can control the properties just by making different volume fractions. And I won't tell you the details, but the important thing to understand, to use as an enzymatic reaction that was first developed, I think it was developed by Peter Schertenberger and when he was in ETH long ago. It's something that people have used for a long time. You use an enzyme and urea and you add salt. So it aggregates very slowly, but eventually aggregates, it forms a gel, and you look at the behavior of the gel as a function of volume fraction. And what you can do is you can measure the elastic modulus, and that's shown here. And notice it depends on volume fraction here as phi to the 3.5. This is 3.5. You can also measure the internal stress. How do you measure the internal stress? You measure the internal stress by when you make the gel, you make it in the rheometer. And so this is a parallel plate, for example. You can use a parallel plate, a Conan plate, and you're oscillating around like this to measure the shear modulus of this gel, but you also measure the force between the plates as it gels, and you find there's a force that's pulling it together. That's why I say it's under tension. And if you measure the value of that tension, it's given by this value. Notice it's less than the value of the modulus, and it has a different slope. It has a slope of 2.3. So how can we understand these things? Well, first, let's understand the rheology. So remember, what's happening is we're starting off with individual particles, and we're slowly making them aggregate to form these larger aggregates, and eventually they gel. So they form these larger aggregates. I'm just drawing it schematically here as a gray dot, but eventually they fill all of space, and they form bonds between them, and they're completely filling space. So how can I understand the rheology of that in the simplest way? Well, look, there's certain number of these things, there's a density of these things. So maybe I just say that if I know that it gels when this length scale is basically such that they fill space, then the modulus should just depend on the density of these. The density of these objects, that should give me a modulus. That's what it always is. It's some intrinsic modulus divided by some length scale, so it's some density. And I can calculate what the density of these things, these are just related to the volume fraction because I'm aggregating them together until the volume fraction becomes 1. And if I ask when does that occur? That occurs when it scales as this relationship, 1 over the fractal dimension minus 3. Well, Stefano measured the fractal dimension. I don't know if I have a plot of that. No, I don't. Stefano measured the fractal dimension. He did neutron scattering, and you see a very nice fractal behavior, and the fractal dimension is about 2.2, which again is consistent with what you know from aggregation of particles under the conditions. This is what we call reaction limited aggregation. It's all consistent with this. So if that's the case, if I don't plot it as a function of volume fraction, I plot it as a function of this correlation length, then I should get an elastic modulus that varies exactly as minus 3. And that's exactly what happens. So it's a very simple dependence on the elastic modulus. What about the internal stress? Well, I can calculate the internal stress. I just say, look, I have a slice across the gel, and I have a certain number of strands running through that slice. And then the internal stress should just be proportional to the number of strands running through that slice. And how many strands are they? Well, it should just depend on the same correlation length, but only the square of the correlation length. And that's exactly what we see. This depends on 1 over the square of that correlation length. This is the correlation length, and it depends on the square. So this is a very simple model that seems to agree with what we see. So we see Internal tension depends on 1 over phi squared. The modulus is 1 over 5 cubed. The yield stress, well, the yield stress is going to be, the yield stress is given by these green. It should behave just like the internal stress, because what we're doing is we're pulling it apart and we have to break a certain number of bonds, and we've just calculated how many bonds we break. It's the same thing. So the yield stress should scale exactly the same way as the internal tension does, and that's exactly what we see. So this tells us now that we have this internal tension, and that's why why the gel pulls itself apart. It's not surprising anymore that we see this additional motion. That's exactly what we're doing. The gel is under this enormous amount of tension. When we break it, it pulls itself apart and moves to the nearest boundary where it's stuck. So everything makes sense. I hope I've convinced you it sort of makes sense, but I'm going to tell you why it doesn't completely make sense, and why there's something that, to be honest, I maybe understand. I'm not sure I understand. I'm going to wave my hands. Maybe I'll convince you, maybe I won't. I'm not sure I've convinced myself, but let me tell you what the real problem is. The real problem is, what I've done is I've calculated everything. I said, look, a modulus is just a density. It depends on the number of bonds per volume. And so I just have to calculate the number of these guys and calculate the bonds for that. And that gives me the exact slope for the modulus, whether I look as a function of this distance or I look as a function of volume fraction. It works just perfectly. And the internal tension just depends on the number of bonds here. So it just depends on the area of this length. So it just depends on how many strands that are going through here. And that goes as -2. And when I put it into the volume fraction, it's 2.3. Again, it's completely consistent. The problem is that if I look inside of here, I also have this sort of network of some sort. And there's probably only 1 strand or 2 strands. There's a small number of strands that pull on each other. But I'm assuming that the modulus, the elastic modulus of this object, is the same at all volume fractions. Whatever it is, it's some rigid unit. And on the one hand, okay, that seems to make sense, and that certainly seems to be what happens. On the other hand, it shouldn't be right, because, you know, as I make this larger, I would have expected to get a weaker behavior. I would have expected, like, it's like saying I have a bunch of particles that are forming a chain, and there's some spring constant between each particle. And now I have a bunch of particles that are all in series. And whenever I add springs in series, the spring constant decreases with the number of springs. So I would have expected there should be something that makes these weaker and weaker each time. And I don't see that. And the only reason I can say that that's the case is that somehow they have to get very, very tough in here, and they have to be so tough that the only bonds that are important are the final bonds that hold these things together. That's where all the weak points are. I'm waving my hands, guys. I don't know, but that's the best I can do. That's what the data tell me. That's what the data support. And that's what it seems to be the case. Let me show you a few sets of data that sort of maybe, possibly may help understand things, but also help me understand something about the origin of the internal stress. So I always used to think, well, if I have an internal stress, if I have things pulling it together, I can understand that I see that a lot in these what are called semi-flexible networks that are just slightly thermal. They're mainly athermal, but they're slightly thermal. And I see that. I've always assumed that when I gel, I have these wiggling arms, and somehow they have to touch, and then they stick to one another. And if it's gelling because of thermal motion, then the most likely thing is not in their natural configuration, but some configuration that's a little bit off. And so they're under tension because I've taken away some of the configuration. So I can understand an internal tension if I have a thermal network, but these are completely athermal. Everything is much, much larger than KT. KT doesn't play a role. So why do we get this internal tension? Where do we get the internal tension? Well, let's look at the time dependence of what we see, and then it gets very interesting. First of all, we see these are just different volume fractions. We see that there's some period of time where if we measure the rheological properties, we can't measure anything. We can't measure a tension. We can't measure everything is within the noise. And then gelation occurs. And what we see is that first there's a very rapid rise in the modulus, and then it seems to saturate a bit and rise more slowly. And initially, there's really no tension. You have to look much longer time before you get tension. So now if I say that it's going to gel down here and I plot not as the zero of time, I don't plot where we start, but I plot where it gels and I subtract that off and I just look at the modulus, you see that there are two power law regimes, one where it's growing very rapidly, another where it's growing much more slowly, just to the t to the 1/2. And you see that the internal tension is also growing. It doesn't really, you see some here, but you don't really see it till everything gets stiff. And then it's growing a little bit faster. It's growing t to the 1/2, 1.2. Here's another one. We see the same thing. It grows very rapidly, then it saturates, and here it grows roughly more slowly. So how can we understand this behavior? It seems like there's something that's happening very fast here and something that's happening much more slowly here. Well, we can go back and we can look at the behavior. If we just look at the long time behavior, there we see that all of the networks, the modulus increases as the square root of time. And that actually has a meaning to us, because what it says is that if you have two particles that come together like this, here you have essentially something that looks like this. And this is going to induce flow. It's basically a very, very high curvature. So you're going to get a little bit of flow of material, and that's going to cause this width here to grow and get larger. And if you just do a simple calculation, and we did this a long time ago, actually it goes even further back. Tom Whitten did a simple calculation of this. It always grows as the square root of time. Let me see if it just grows as the square root of time. And that's exactly what you see here. And we've seen this kind of behavior before. So the long time behavior is because this is getting larger and larger. If I go back here, that doesn't account for this behavior. My picture for this, and this is just, again, a bit of a hand wave, but it's how I understand it. This is where it first gels. And the reason it's gelling is you're getting the final formation of a connected network, but you still, even though you have like something that looks like this that's connected, you still have some other particles, some other clusters that are diffusing around, and they can join and they can form, they can join places where you take all the weak bonds, you have new bridges, so you get like things that will strengthen things. So I would like to my analogy is, if I turn it around and if I were weakening it by cutting bonds, that would be like elasticity percolation. It would be some very, very strong dependence on the number of bonds. I'm just adding some strong bonds. I'm looking at the other way around. I'm looking at it as a function of time. But that to me is why, up to here, you're getting really rigid regions, because you're getting things that strengthen them, but still you're left with the final bonds where you have these weak bonds between the particles, roughly one per cluster. That's my argument. And that's consistent with why the whole thing strengthens over long times, because there you're just making the bonds more rigid. You can ask other questions. For example, if that's the case, if this is getting thicker, if this is growing as the square root of time, how is the distance between them. How is this distance growing? Well, it's related by the square. So this is actually growing as the square of time. So, the square of this length, which means that it's growing linearly with time. And that's exactly what we see. The internal modulus, if it's just where the gel is pulling itself in like this, because this is getting narrower, it's literally collapsing on itself, that grows t to the 1. This grows as t to the 1/2. It really is consistent with the kind of dependence that you would expect for a network like this. And if you look at this, you can make some more predictions. Look, this is the slope of the particles I show. We can change the particle size. Remember, everything should scale basically as the size of the particle. And the modulus should do that. The other one should scale as the area. So it should scale as the area of this region. That should be as r squared. So if we look, how does it depend? How does the modulus depend on the size of the particle? Okay, we've just done a couple of particles. We don't have a lot of data, but it scales as the area. So again, it's consistent. I don't want to say that it proves it, but it's absolutely consistent. So what does this tell me? It tells me that now I really think I understand something about the nature of these gels. Let me go here so I can draw. What I'm saying is that I form these networks. The networks coarsen. They coarsen because this is growing larger. They're coarsening like this. This is getting larger, and this is consistent with it getting larger because the area is growing and it's proportional to the radius of the particle squared. It's getting larger because it's coarsening, so it's growing. It's coarsening as the square root of time, consistent with that. With what we see, we would only have looked and seen this internal tension having done these DSH measurements where we saw this extra motion, and then we went and we measured the motion in the third direction, and it's really literally moving in the third direction, pulling itself apart, coming closer to the boundaries. So with that, I hope I've convinced you that we have a cool new technique that lets us do a lot of interesting analysis of things. We can look at an old problem that's colloidal gelation, and we see that really it's not that we're fracturing it. We're not doing fracture by driving some fluid through, but we're doing fracture by pulling water out. And as we pull water out, it just breaks the last little bond, and it literally tears itself apart because of the internal tension. And with that, let me thank you, and I hope there's time for questions if anybody wants to ask them.
So I'm just going to dive in real fast and ask about something completely different from what you've told us. So have you applied your this new dynamic speckle holography to bacterial swarms? Because you could look at swirls on really huge systems now.
Yeah. No, we haven't, David.
Because bacterial swarms, biofilms, the whole 9 yards. Everyone has been stuck because they're looking at tiny little regions of a really big, complicated process. This could unlock those problems.
You should really pick up the large scale swarms. Absolutely. It's a good idea. Great idea.
Dave, do you need single scattering, or does it work with multiple scattering?
You need single scattering for this.
Yeah, that's what I thought.
Yeah, I don't know how to make it work with multiple scattering.
So you can't use it on skin and stuff like that.
Well, so one thing I've learned is that you can get single scattering for a little bit more than you think. So it's like we always know that the first few layers, if you can do single scattering. So if you do the analysis, I don't know. I would like to ask whether, particularly in backscattering, whether we can do so that just it's scattered once or twice, we still may learn something. I can't say.
So Dave, I mean,
Yeah.
this technique is what we used to call homodyne technique, in that you're interfering light scattering off of different scatterers.
That's heterodyne, Dave.
No, that's homodyne.
It's heterodyne if you use another beam, right?
Yeah. Jesus, it's been so long. You're right.
So, yeah, so
Yeah, you're right.
the thing is, you could imagine, I mean, it's going to depend upon the system and the problem you want to look at. But what you end up looking at, which you, as you pointed out, is really differences in velocities of particles. You're looking at strain, if you will, in some sense.
Yeah.
But you could do a real heterodyne measurement if you have a transparent sample, you shine the light through, and you interfere the scattered light with the unscattered light. Then you get the absolute motion of the particles. It's the same thing. Have you thought about doing that kind of thing? Because then you're measuring actual velocities of individual things.
Yeah, no, we haven't, because what we're doing, I mean, so you're right, Dave. The thing that I brushed over is that we don't know the direction of the motion. We just know it's parallel.
Yeah.
And so we have to add physics to get the direction of the motion. And there's a lot of additional analysis that I didn't tell you to take advantage of. You know that correlation at one time difference and two time differences have to add to the third time difference if we're seeing physics, if we're seeing strain fields. And so we have to do all those tricks. But you're absolutely right. If we really did true heterodyning, then we would not do it. You could also, it's like doing when you get the phase measurement in X-rays, you put some heavy atom to give you extra scattering. So you could do the same thing. You put some stationary object or some big scatterer that would.
Yeah. Peter Pusey used to do that. That's the way he used to do his heterodyne measurements. You could do a riff on that.
Right. Well, Dave, this is just a riff on everything, right? It's it's just summing up all the different ways that we've done scattering over the years, but doing it slightly differently. But that's a good idea.
I have a quick question about this internal tension that you're measuring. So it seemed to have this like area dependence, right? It had this what was the square of the volume fraction or something like that. Is that a result of the geometry of your setup, where you have it in on a slide? Like if I had a ball of colloidal gel stuff and I'm drying it, does that scaling look different?
It's geometry, but slightly different. Iraj, it's can you see my fingers? So you see the spheres. It's the fact that they're joined right in a perfect figure eight, and then they're coming together like this, and they're flattening out here. So it's how much it flattens out, and that scales as the size. It scales as the area. So it scales as the radius of the particle squared. So that's the geometry. So it's not the macroscopic geometry, it's the geometry of the sphere itself. And so it scales as the radius of the particle that makes up the colloidal gel squared.
Thanks.
What scale, down to what scale can you measure strain? I mean, can you measure it inside a cell?
Yeah, sure. Because, well, okay, it depends. It's a combination, Paul. It's a combination of how fast it goes and how sensitive you are. So if it's going slowly enough, then you can watch a speckle pattern change over longer periods of time. And then you can see, it's just a tiny decay, and you can basically add them up. If it's going really fast, so you don't have the temporal resolution, then you're limited to how much you can trust, basically how much the intensity changes. So I don't know, if it changes a half, then it's a half a wavelength. If you see a tenth of it, it's a tenth of a wavelength, something like that.
But still, it's sort of limited by the wavelength. That is, you're not going to see strain inside a molecular motor or something.
Probably not, because the strain will undergo lots of changes, right? If it were a strain field that that changed over time in some linear way, you could probably measure well under a wavelength, because you can measure, maybe a tenth or a hundredth of a wavelength, even though you only see a tiny change in the correlation. You can add those up by looking at different time, T1 minus T2, T2 minus T3, and then T3 minus T1 to do a back check and do all that correlations. And then you can see the strain, but it has to be uniform strain. You have to make the assumption that it's uniform or roughly uniform in time. If it goes back and forth, you won't be able to see it.
I mean, you can measure tiny strains, but you still have to resolve where they're coming from, right?
Yes, that's right. That's right. So, right. If you're looking at a tiny object that you'd need to have enough light in there to contribute significantly to a speckle, and a motor is unlikely to do it.
Dave, when you push this water by the front of air and pressure in water is below pressure in air, water presumably evaporates through this curved front. Is there a balance of masses of water?
I don't think it's that. There's a balance of mass, but I don't think it's that, because I think that the evaporation rate, then it will evaporate, but you'll immediately saturate the volume where
Of
that
air. That was my question, yes.
Yeah, then you'd have to diffuse all the vapor out, and that's just too long. Instead, what I think is happening is that it's being transported along the interface of the gel, because it's all drying. And so it's been transported as a thin layer of water, either along the glass that's been broken off, or more likely along the interface of the gel. So you really have transport of water, literally flow of water, because of capillary effects, all the way back to where you can dry it at the surface, where it gets out. That's what I think. Yeah. Otherwise, it's just too slow. It just wouldn't work.
And there is room for water to flow, because I didn't understand where water goes.
Well, yeah, but remember where the air is, it's really literally a crack, and that crack, that crack goes all the way out to where the air is coming. So it's running along the edge of that crack.
Okay. And do you see this flowing water?
No, I haven't measured. It would be we could try, but we haven't tried that. I'm only doing sort of back of the envelope calculations to tell me this.
And your envelope is too small to see this water, right?
My envelope is too small. No, my envelope is I haven't done enough experiments. You're telling me all the experiments I should do, and you're right. I should do them. I should just try that. We haven't done that, but we know that that's something to do. We a lot of the experiments that we do for just because the way what we were doing, and before we understood anything of what we were doing, we were actually injecting oil. So then the physics is the same, but you don't have to worry about it. You have to actually worry, where does the water end up? Where's the excess water? Because you're putting oil in, there's the water. And if you look carefully, again, we did it only after the fact. We look carefully back at the entrance. There's some water that's being collected. So there's some propagation. But you're right. Conservation of volume. You can't get around it.
Conservation of mass, anyway.
Yeah. Conservation mass. Right.
Knowing you probably already thought of the killer app. What's the killer app?
Well, for me, these measuring these strain fields is really, really interesting.
Yeah, measuring strain fields is great. But I mean, knowing you probably have already figured out that this is going to cure something, or it's going to revolutionize something. Right?
Not yet. Well, I was talking to Alexandra's students before this, and they were asking about this, and I was saying, geez, can't you do it in the nucleus and watch the chromatin? Could you do that, Alexandra? I don't know, maybe not, but it would be so cool if you could.
I'd love that.
It would, absolutely.
Yeah. I don't know. Yeah. So I don't know. I don't know the killer app for this. Well, what was the
Well,
killer app for
in any
this?
living systems, right? I mean, if you basically start to look into any living systems, right, either starting from the large scale, the biofilms and colonies that David proposed, to then going inside of a cell, and I mean, those will be fantastic readouts.
Yeah, always the question is, can you get speckles? Do you get enough scattering from what you want to see moving that it contributes to the speckle? And that's what Paul's point was.
Yeah.
It would be more difficult.
Maybe in vitro systems, though.
Yeah.
Right. So they're. And
Well, yeah, no, you're right. We've suddenly gotten interested in these phase separating regions, and so we're doing microreology, but maybe for
cool.
microreology.
Very cool. Yeah, and also in vitro, right? You could basically do this enriching in like heavy elements as you propose, as for anomalous scattering people do, and in vitro that would actually work very cleanly, very nicely.
Yeah. Yes. Good point. Good point. Thanks for trying. As always, great.
So what do you guys look now into the liquid phase separations in the liquid condensates?
I don't know. Do you know Thomas Knowles?
Thomas Knowles. How do you write Knowles?
At Cambridge.
Okay, of course I do. Yes, of course. That's why I had to remind myself it's with K.
So my stupidity, Cliff Brangwyn discovered these things, and I said, well, I gotta let. He was a student of mine, so I said, I gotta let Cliff do his own thing. So I said, I'm not gonna do any of this. And then 10 years later, Thomas comes to me and says, you got to do this with me. You got to do this. He got me all interested. And his motivation is like Paul says, what's the killer app? What's really the killer app? And here's his argument. Look, what's the paradigm, the basic paradigm of biology? DNA to RNA to proteins. How do you treat disease? Modern treatment of disease. Proteins, you try and use antibodies to change the way proteins behave, the signaling pathways of proteins, or you use RNA to make a vaccine, or you use CRISPR to change the way the nucleus does. So you're always trying to drug the basic paradigm of biology. But now here's a new paradigm. At each level in the nucleus, you've shown that there are these droplets that form and that somehow affect things. In the cytoplasm, you see these droplets that form that involve the RNA. You see the droplets that form that involve the proteins. So maybe you don't drug what you traditionally drug. You drug the droplets. You drug the phase separation. You drug the phase behavior. A completely new way to think about drugs.
Exactly. You could start to move within the phase diagram.
Yeah, that seems like something that we ought to do. Can't not do that. So that's what we're doing.
Very cool. Very cool.
Set up a company.
We did. We did.
You probably did. Yeah.
We did. It's in my lab already, Paul. I mean, you can't, it's too big to do by myself. I can't do it myself. And we're not alone. We're not the only people who thought of it. But absolutely, that's what we did.
So next year you go to a doctor and doctor prescribes you, Dave, you should push from liquid phase to pneumatic phase.
Exactly. But the fun thing, Paul, is that that's the motivation, but there's so much really cool physics. We have this weekly meeting where I think like it's like your place. 5 professors from Harvard professors come to a Zoom meeting, and we all learn about these liquid condensates. It never happens at Harvard. It happens at your place. We're learning from you. It's great. It's fantastic. I love it.
Better without Zoom.
Yeah.
Well,
soon out.
The only thing is that that's the one thing that makes everybody around. So
It's true.
You got to look. I don't know, spent a whole year. Got to look for the positive things. That's one of the positive things.
Yes.
It leads to a lot of cool new physics, man. A lot of really cool new physics.
Yep, something new. Great.
So is the camera super expensive because the camera is super sensitive or because it's super fast? What makes it expensive?
It's not that expensive. It's just the most expensive part. It's we didn't have lots of cameras. Ideally, what you can see what you ideally want is you want 3 lasers and 4 cameras, I think, to do this properly. But we didn't have that much, so it's not. We don't use a super. We should use a very sensitive and very fast camera. The fastest camera you can get away with. And that's just limited by the amount of scattered light there is. I can't tell you exactly which camera Stefano used. I don't remember. He's one of the high-speed cameras.
Cool. So when do we get you down for dinner?
As soon as you're open, I'm there, man. I'm ready to fly. I'm done. I'm finished.
What do you mean fly? Aren't you going to drive?
I drive. I do anything. Sure. You tell me when it's open, Paul. I'll come. I'll come and have dinner with you.
Now you're talking. Sounds good.
So,
Okay,
Should I stop recording at this point so we go offline?
sure.
Let's do that.
You're host, right?
I believe